Recording in progress.
I was telling you the function of the glycolipid. Firstly, I was telling you the difference between the ligand and a receptor. When we talk about a receptor is anything which you can say that respond to something that is, just like this. The glycoprotein over here. This glycoprotein is a receptor molecule. And you have done this in IGCSE in the coordination chapter. That what is the difference between receptor, stimulus, and effectors? So receptors are those which detect any of the external stimulus. Basically, in biology, we say receptors are those components, are those compounds present in our body which respond to some of the external stimuli. Okay, they respond to the stimulus. And what is the stimulus over here? The ligand is the stimulus over here. And it may be hormone, it can be neurotransmitter, it can be drug. So whenever a drug or a neurotransmitter is approaching cell membrane, so within cell membrane there are receptors which will respond to that particular type of neurotransmitter. And those molecules are known as receptors, the glycoproteins. Are you getting the difference between receptor and ligand? Receptor is something which will detect a stimulus. Basically, simply when we talk about receptor is something that will yes, I have written over here that it is a specialized protein present outside the cell surface for for detection of external stimulus. This is what we say a receptor is in biology. So the why I am saying that glycoproteins are receptors because they detect some hormone or neurotransmitter or the drug when those drug or neurotransmitter approach them, when they approach the cell membrane. And those neurotransmitter and hormones and drugs are ligands. So I hope you are clear with the difference between ligand and a receptor.
Yeah.
These are two different things. Okay. Now, yes, we were discussing the role of glycolipid and glycoprotein. Basically, I have written that the glycolipid and the glycoprotein they act as receptor molecule. Now, what is a receptor molecule? Which will detect,
Receive a
yeah,
signal.
which will
Detect
detect.
a signal.
Hmm. So we can write it over
A
here.
signaling molecule.
Yes, detect stimulus. That is what is also known as signaling molecule. So the glycolipid and glycoproteins are known as signaling molecules, and they bind ligand molecule with them. And what can be any ligand? It can be any hormone,
Hormone,
hmm,
neurotransmitter,
or
and
any type of
drugs.
drug. For them, you have specialized type of receptors in the cell membrane. Clear?
Yeah.
And this is the function of them. Now, okay, can you tell me the difference between receptor and the stimulus?
Stimulus?
A receptor and the ligand. Sorry.
Ligand?
Mhm.
A receptor protein is something that receives the signal, and a ligand is a signaling molecule.
Which one? Yes. Okay. And when if they ask you the role of glycolipid and glycoprotein, what you will say?
They act as ligand molecules which bind with external
No, they
stimulus.
are not ligand molecules. They are receptor molecules.
Yeah, they're receptor molecules
Hmm. They
that
will
bind
bind.
with ligand.
They will bind
They
ligand
bind.
with themselves. Okay. Whenever a
Yeah.
ligand will approach the cell membrane, those glycolipids and glycoproteins they will serve as a receptor molecule, and they will detect that ligand and they will bind that ligand with them. Clear?
Yeah.
Okay. Then we have the role of the glyco, what I was saying, the glycolipid and glycoprotein. And one more thing, when we talk about the receptor molecule, that they are receptor molecule, you have basically three type of receptors. Okay, in
Okay.
the cell membrane. within the
Three
cell
types
membrane.
of receptors.
Yes, types of receptors in cell membrane. The first one is pen is not working. Okay, the first one is signaling receptor, and these are we have uh, discussed that they will provide signal for forward for neurotransmitters or
Hormones.
yes, and hormones. This is the first type. Then there are some of the receptors. They help in the stabilization of the cell membrane. We have discussed about the stabilization. What is meant by stabilization of the cell membrane? In the last lecture, when I was telling you about the cholesterol, that the cholesterol maintain the fluidity of the cell membrane. Otherwise, it will be cell membrane will be destabilized. So there are some receptors as well. Those receptors will also help to stabilize the cell membrane. Okay, so such type of receptors, they involve in the stabilization process. I can write it over here. Receptors for cell membrane stabilization. And how these type of receptors work. Just like we have discussed the function of different things, if they ask you about the receptors which are responsible for the stabilization of the cell membrane, and they ask you how they work, so you must know that when we talk about the stabilization, that these type of receptors help in the stabilization of cell membrane by adhering some of the substances on them. Are you
By
getting this?
what?
The concept of cell adhesion of some of the important things. I'm writing it over here. Cell
No.
adhesion. Adhere. What is adhere? Adhere means to stick something over it. To stick something over the surface. So such type of receptors work like this, that they bind some of the substances on the cell membrane. So this is the second type of receptor that help to
So
they bind some of the substances
they
on
bind
them. They basically these receptors are present in the cell membrane. Okay, and within the cell membrane, if some of the specific type of the molecules can bind to these receptors, that's why these receptors are known as they are responsible for cell adhesion, and they help in the stabilization of the cell membrane. Just like, for example, you have a cell, and around the cell there is a carbohydrate molecule. Okay, so how that carbohydrate molecule can adhere on the cell membrane? Which part of the carbohydrate can attach?
it can attach to
Mhm.
the membrane by the receptor.
By in the carbohydrate there are hydrogen bonds. We have discussed this. If you remember the glucose structure of the
Yeah.
glucose, there are hydrogen bonds, right? So by those hydrogen bonds, that carbohydrate molecule will connect with the water present within the cell. Are you getting this? Because there is aqueous environment. Okay, let me draw this. What I am saying. I am saying
So it so the carbohydrate molecule binds with the water.
yes, within the cell. Like for example, you have
Okay.
a cell just like this. Okay, and outside the cell you have a glucose molecule. Okay, let's say this is a glucose molecule. Over here there is oxygen and all these things. How this molecule will attach? You know, it has hydrogen bonds. So those hydrogens will combine with the water present over here. Okay, or water molecule surrounding the cell. If you know the aqueous, this side.
So
You
it
know the
binds to the water. So
yes,
it forms hydrogen bonds with the water on like on the surface of the cell
yes.
membrane.
We have discussed that there is phospholipid bilayer, right? That's why
Yeah.
outside there is hydrophilic heads, and inside as well there is hydrophilic head, and in between you have the tail region. You get this? So the carbohydrate molecule, the hydrogen bonds, they will stick to this part. They will stick to the outer part of the cell membrane. This is what we say adhesion process. Adhesion means to stick at some place. In Urdu, we say chipakna, like you have something and that is to stick.
So
If you, huh?
it sticks to the phospholipid bilayer.
Yes, exactly. And then after, because you know that glucose molecule has to come inside the cell, but it cannot directly come. You know, we have discussed that it needs to modify because glucose molecule is a large molecule. So firstly, it will stick, and after that there will be modification, and that modification will be done by the carrier protein, and then that glucose molecule can enter within the cell.
Okay.
So these are those receptors which help in the adhesion, and they also help to stabilize the cell membrane. Otherwise, you know why I have mentioned the stabilization over here? Because otherwise, if a glucose molecule is approaching a cell, what can happen? Maybe it can change the configuration of the cell membrane. Like it can try to enter within the phospholipid bilayer, and it can change the structure of the phospholipid bilayer. So such type of receptors keep the cell membrane stabilized. They don't allow any changes in the cell membrane. They will
Okay.
allow the adhesion process, and some of the substances can stick to it. And yeah, these are those receptors which will help in stabilization. And the first one was the signaling receptor that will provide the signal to specific type of ligands, just like neurotransmitters or hormones. They all of these are present in the cell membrane. And the third type of receptors are involved in the endocytosis. You know endocytosis?
Endocytosis. What?
Hmm.
Endocytosis?
Endocytosis, yes. Now you cannot say you didn't study it in the IUCSE. Huh?
I've heard of it.
Huh? You forget that, right?
Yeah.
Okay. There are two things, endocytosis and exocytosis. Let me write it
In
first. Mm-hmm.
immunity.
Yeah, think about it.
Oh yeah. It was like the immunity.
No, no, no. It is
The
not related to immunity. It is a
no.
type of transport. It is uh, in the category of the transport, the active transport. It is an active transport. Basically, endocytosis is the process in which the cell ingests some of the large molecules by engulfing them. This is known as
Now
endocytosis. Engulf. Yeah.
it
I
engulfs the
yes,
oxygen.
that is endocytosis. And what is exocytosis?
It's when it releases the substances
Yes.
which attack the
Hmm.
microorganism.
Yes. Just like, can you give me an example of exocytosis?
Like
Mm-hmm.
attacking the pathogen by
Yes.
using
Mm-hmm. By
by
using T cells or
yeah
Okay. Endocytosis is an active transport mechanism in which the cell, yes, it ingests large molecules by engulfing them. It will engulf first, and then there will be ingestion, engulfing them in plasma membrane. In this fluid mosaic model, the membrane in the plasma membrane. I'm writing the definition over here, and then I will tell you what I have written. Which will then, this plasma membrane will then pinch off this large molecule when it will be entered in the plasma membrane by engulfing process. After that, it will be pinched off into a vesicle, in a vesicle form. Now, what does it mean? It means like you have a cell just like this, and maybe this is a large molecule. This is a large molecule, and it want to enter the cell. Firstly, it will be adhered over here, right? And then it will pass through the plasma membrane through some of the carrier protein. And when it is passing through the plasma membrane, it will be punched off in the form of vesicle. Okay, in the form of vesicle.
What? It will be
I'm saying
transported in the form of
vesicle,
vesicle?
small vesicle.
Okay.
Firstly, it will adhere, then there will be modification, and after that, when it is on the surface of the cell, after that, when it will be entered in the cell, it will enter in the form of vesicles. So this process is known as engulfing. That cell is engulfing something, and when it will engulf this particular molecule, it that will be pinched off. You know, this is known as pinched off. That firstly, it is over here, right? Now it will connect to the cell membrane from the inner side, and then it will convert into a vesicle form. Are you getting this? What I'm saying?
Yeah.
Just like you have, yeah, when you pinch this off, now in this will be, you have this form, now inner aberration, and this will now convert into a vesicle. So this is what we say endocytosis. So there are receptors that are on the cell membrane which are involved in the endocytosis. Otherwise, if you know those receptors, as we have discussed, that cell membrane is a semi-permeable membrane. If there will be no receptors available, then what will happen? Every type of molecule will enter the cell. So these receptors are checkpoints. They will provide the partial permeability to the cell. Otherwise, the cell membrane can be fully permeable on these receptors and on these glycoproteins and glycolipids and the all other conjugate molecules, on the behalf of these things we say that cell membrane is partially permeable. Otherwise, if you don't have these things, if you just have phospholipid bilayer, then that phospholipid bilayer can allow some of the like the lipid soluble things to pass through, and it cannot allow the water soluble things to pass through it, or sometimes it will.
It doesn't let the polar molecules pass through.
If I'm talking about, if there is just phospholipid bilayer,
Yeah.
I'm just giving you the example. I'm
Yeah.
telling you the importance of all these things, all these conjugate molecules. All these conjugate molecules collectively give the partial permeability to the cell membrane.
Yeah. Okay.
Okay. And the endocytosis can be of two types, phagocytosis and pinocytosis. I'm just writing the name and you know the things, right? You know?
Endocytosis and phagocytosis. Yeah.
Are you sure?
Yeah.
Okay, great. Exocytosis and endocytosis. Okay. Now after this you have yeah, these were the receptors which we have discussed, and one more type of receptor is there. Actually, there are four types actually. When when there are just like you were telling me about the immunity, when there are diseases in your body, when any pathogen enter your body, so your body releases antibodies against that particular type of pathogen. So there are some of the receptors as well, which will help to recognize the antigen. Okay, they act as the antigens, and they will respond to that particular type of pathogen. Are you getting my point? They will help in recognition of the
Yeah.
pathogen. So there are receptors that will help in cell recognition. What type? Recognition of
Recognition of
hmm, pathogens.
microorganisms.
Yes, and these type of receptors, they will act as they act as antigens. Okay, are you clear with this?
Yeah.
Recording in progress.
These are the some of the important types of receptors present in the cell membrane. Can you tell me the names of these receptors?
All of them
The type of receptors here in the cell
on
membrane.
the cell membrane.
Mm-hmm.
There's signaling receptors
Mm-hmm.
that receive the signaling molecules like
Hmm.
neurotransmitters and
Yes.
hormones. And then there's receptors for cell membrane stabilization. This is um, this is like for the adhesion, cell adhesion,
Yes.
for like glucose and like the larger molecules to adhere to the cell membrane
Hmm.
and enter the cell
Yes.
by the carrier protein
Yes.
on on the cell membrane. And then there's receptors that are involved in endocytosis. Endocytosis is like when a cell takes the substances into itself, engulfs it. It's an active transport mechanism in which
Okay.
cell like ingests large molecules by engulfing them in its plasma membrane,
Okay.
which will then pinch
Hmm.
off into a vesicle. It will change into a vesicle.
Yes.
The large molecule, when it enters the cell, it will be in the form of a vesicle. And then there's receptors which help in cell
That
recognition of
molecule
fat.
can enter in the form of a vesicle.
Yeah.
Okay.
And then there's receptors that help in cell recognition of pathogens.
Hmm.
These act as antigens and recognize when a pathogen is in the cell or near
Yes.
the cell.
Yes. And last but not the least, the proteins. What we have discussed about the proteins. We have discussed the role of the proteins. There are
Yeah.
two main type of proteins. What are those?
The from the start, globular and fibrous.
No, 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 no.
On the there's glycoproteins
No.
or
Functions. On
carrier
the basis of
proteins.
functions.
Yep.
Role of proteins. On the basis
Channel
of function,
proteins
you have,
and
yes, exactly. You have two types of proteins. Those are, first one is
channel proteins
hmm, carrier protein and
and channel proteins.
channel proteins. Can you tell me the difference?
Carrier proteins have a site that is complementary to a specific substance. It binds with the substance and allows it to enter the cell. And channel proteins, it creates a
Mm-hmm. It serves as a
it
gateway.
creates a
Mm-hmm.
hydrophilic acid for the molecules to enter the cell.
For just like the ATP, or not ATP, it will the sodium ion or potassium ions.
Yeah, the ions to enter the
Yes.
cell through
So
the
the basic difference
hydrophilic
is that
acid.
the carrier proteins, they will on the carrier protein, when we have discussion about this, you will say that they will bind molecules with them, and then they undergo
Change,
conformational changes in them.
yeah,
And
the
what are
shape of the
conformational
carrier
changes?
protein.
Yes, they will change the
It's yeah,
shape of the molecule,
to transport the
hmm,
substances across the membrane.
and release them in the cell.
Yeah,
Release them in cell. And the carrier proteins, when we talk about the carrier proteins, in comparison to channel proteins, they are slow in their working. So I am just telling you the difference. So firstly,
okay.
this is second, they are slow as compare in comparison to channel proteins. And you can tell me why they are slow.
Because it takes time for it to
Hmm. Yes,
undergo the
to
conformational
modify it,
change.
to modify the
Yeah,
substance. The when you have the conformational changes, you can also say the modification of the molecule.
okay.
Okay, and mm, this is, you can say, a carrier protein is, the example is glucose transporter protein is a carrier protein. Okay,
Okay.
and yes, they are very specific. They are highly specific. Like for glucose, you will have a specific carrier protein for glucose, and for other molecule you will have the specific with regards to that. But when we talk about the channel protein, they can allow sodium, potassium, calcium at the same time. So they are specific, highly specific.
Okay.
Okay. Then you have channel proteins. And what is the function of the channel proteins? We say that channel proteins, they
Create a
they
passage
create a
or
gate. Hmm.
a gate.
Yeah. Through the phospholipid bilayer. And they are as compared to the carrier protein, they are fast. Okay, because they are just transporting the things fast. Rapid movement will occur of molecules occur. Then they are not specific. They are not highly specific. You can say that, but when I talk about the carrier proteins, they are specific in terms of the biomolecules. But when we talk about the channel proteins, you will say they are highly specific for ions. They will allow ions to pass through.
Specific ions.
Yes, the sodium, potassium, and calcium. Basically, you will have sodium and potassium pumps. You will see in the upcoming chapter. highly specific for sodium and potassium ions. Okay. I hope you are clear with the difference of these and what are the role of the proteins present in
Yeah.
the membrane. And the transport these proteins, when we say that the and how these proteins are present in the cell membrane, you will say that these proteins, if you see this diagram over here, these are present throughout the cell membrane, right? Within the cell membrane. So you can say that
Mm-hmm.
they create the channels, they create hydrophilic channels basically. Because
Yeah.
some of these, because otherwise, if you see the water molecule cannot enter from these regions because there is fatty layer in the center. So simply we can say that the proteins, these transport proteins, they produce hydrophilic channels for the molecules to pass through. It will allow polar molecules to pass through it. Okay. The basic function of the protein, if they ask you in particular, so you will say that they create hydrophilic channels and then they will allow polar molecules to pass through. Polar and ionic substances. Clear? The proteins, they create hydrophilic channels. Otherwise, the water molecule or the polar molecules cannot pass the phospholipid bilayer because in the center of the phospholipid bilayer, you have fatty acid chains within the center. So those fatty acid chain will not allow water to pass through. So if you want water or polar molecule or ions to pass through the cell membrane, you need some of the important channels, some of the specific channels, and these are hydrophilic in nature. They will allow polar and ionic substances to pass through. And all the non-polar things can easily diffuse from the cell membrane. Why? Because the major portion of the center is made up of fatty layer. If you see this. Are you clear with what I am
Yeah.
telling you? Okay. And yeah, this was about the transport proteins and the carrier proteins. Yeah, one more thing. When we talk about the transport proteins, they will allow, as we have discussed, that they will allow some of the substances to enter and some of the substances to leave. So, what should be your suggestion about the number of these proteins? How many these proteins, these transport proteins, should be in the cell membrane? Because they are responsible to control the substances, the entrance and exit of these substances. So, how much they must be
How much they must be
in the cell
like
membrane?
throughout
The
the cell?
transport proteins, yes, not in the cell in the cell membrane.
Cell membrane.
Mhm.
They
Because they
must
are
be.
controlling the substances, the entrance and exit of the substances.
It should be throughout the cell.
Yes, it should be throughout
Cell
the cell membrane.
membrane.
Exactly. Because within the cell, the substance can try to enter from different position. Okay, it is not mandatory that this is the gateway for the glucose molecule and this is the gateway for lipid to enter. They can approach the cell from any side because there is blood and in blood you have everything. So that is why throughout the cell membrane, transport proteins should be there and because they will control the exit and entrance of the particular type of substances. And what can be those substances? Which substances can pass
What?
through the proteins?
To the carrier proteins.
Mhm.
Um,
In
like
general, in general.
like glucose, like glucose,
Mm. And
and like
polar.
non-polar.
Mm. No,
Molecules.
non-polar substances
Fatty
don't need
acids.
any channels. Non-polar substances don't
They
need
have
any
channels
proteins. Okay, okay.
to carrier protein. The
I am
polar
asking you the role of the proteins. Which substances they will allow?
um they will allow the proteins will allow ions.
Hmm. And
They
what is
will
this?
allow what is one?
This one.
Polar and ionic substances.
Hmm?
Polar and ionic substances to enter the cell.
Yes, ionic and polar substances. Because
Yeah.
non-polar substances
Can
can easily diffuse. They don't need any channels. They don't need any specialized channels. But
okay.
the polar substances need specialized channels, and I have told you the reason why they need specialized channels. Because in the center there is all this is fatty layer, right? This is
Yeah.
fatty layer. This will if any water molecule want to enter it, so it can pass. The water molecule can pass this, okay, but it cannot enter over here. It cannot pass this because the
Yeah.
oil, the fatty layer is non-polar in nature and water is polar in nature. So that is why you need such specific type of molecules which will allow water or polar substances and ionic substances to pass through the cell membrane. And all the non-polar substances can easily diffuse. Clear?
Yeah.
Okay. One more thing, when we talk about the fatty acid, like there are fatty acid. I told you in the when we were discussing the lipids that there can be saturated fatty acid in it and there can be unsaturated fatty acid in it. You remember? Or should I repeat that?
Yeah, there's unsaturated and saturated fatty
Yes,
acids.
yes, exactly. So in the lipid, there can be saturated fatty acids and there can be unsaturated fatty acids. So the
Yeah.
fluidity depends on it, basically. When we talk about the fluidity of the cell membrane depends on the whether you whether there is more saturated fatty acid in the cell membrane or whether there are more unsaturated fatty acid in the cell membrane. So it depends on the fluidity of the cell membrane. Remember this line, the fluidity of the cell membrane depends on the composition of the fatty acid chain present in the phospholipid bilayer because the fatty acid chain can be saturated